So, as nobody has joined till now, I will just start the session in 10 minutes. Good evening, everyone. Welcome to the week 6 of Biomass Conversion and Biorefinery. The course coordinator for this course is Professor Kaustubh Mohanty from the Department of Chemical Engineering, IIT Guwahati, and I am Arihant Ahuja, a PhD scholar in the Department of Paper Technology, IIT Roorkee. I am currently working under the guidance of Professor Ubhar Kumar Saraswati, who is in IIT Roorkee, Department of Paper Technology. Before moving on the week 6 uh, doubt session, first I would like to tell you about something about me that I have done my BTech in chemical engineering from Bipin Tripathi Kumar Institute of Technology, Dorat, and I did my MTech from IIT Roorkee and currently pursuing PhD program in IIT Roorkee. And I'm receiving Prime Minister Research Fellowship from 2021 on the topic of paper-based value-added products. So today we will be discussing about the week 6 assignment and week 7 assignment. We will discuss the week 6 assignments from 2021 to 2023, and then further we will have a discussion on week 7 assignments of 2021 and 2022, so that you could have help in week 7 assignments of 2023. So we will start from the assignment 6 of 2023. Uh, you all might have submitted the answers to assignment 6 2023, and some of you have might have submitted the right and some might have done it wrong. So here I will tell you that you may also know the right answer, but here I will tell you that what is the theory behind that answer, and it will also help you in revision of your coursework. So let's move on to the first question. Following which process is not considered as microbial conversion process? Fermentation, anaerobic digestion, composting, or pyrolysis? So the main aim of this question is that we have to find if one of these is not a microbial conversion. So fermentation involves yeast. We all know that fermentation involves yeast. A typical product which we use somewhat in a daily life is bread. Bread is made by the process of fermentation, and yeast is used in it. Anaerobic digestion is a process in which we use microbes. So first was yeast, and second we use microbes under the absence of oxygen. That's why its name is anaerobic. And third is composting. Composting also involves some kinds of microorganisms, and it is used to convert biomass into compost, which can be used in soil for further growing of plants. So these three, the first three involve some type of microbacteria and yeast. So these three are typical microbial conversions. But the pyrolysis method is not. Pyrolysis is a thermal process. Pyrolysis method involves heating of heating of biomass in absence of oxygen, and it can be under vacuum also. So pyrolysis is a thermal process, and rest three are microbial processes. So the right answer for this is pyrolysis. So before going to the next question, I will also explain that what are these processes and how all these are different from each other. So first we'll move on to the pyrolysis, as this was the answer. Pyrolysis is not considered as a microbial conversion process, as it involves the thermal decomposition of organic material in the absence of oxygen without the involvement of microorganisms. So no microorganisms are involved. In contrast, fermentation, anaerobic digestion, and composting are all microbial conversions. The rest were microbial, and they rely on the activity of microorganisms to break down organic materials into useful end products such as alcohol, biogas, and compost. So the diagram which you are seeing here is a typical pyrolysis reactor, and the pre-treated biomass is fed to the pyrolysis chamber, and combustion process takes place. So heating is done in the absence of oxygen, and we get what is known as raw produced gases. So this is a typical pyrolysis process, and it does not involve any type of microorganisms. And if we look at the other methods which were mentioned in the option, first was fermentation. So during fermentation, microorganisms such as yeast and bacteria break down organic compounds such as sugars and produce useful products such as ethanol, lactic acid, and acetic acid. So it is a uh, phenomena to break down the sugars into smaller components. So if we treat the sugars with the yeast, then we can get some gases such as CO2 and alcohols, particularly ethanol. So fermentation involves some kinds of microorganisms. During anaerobic digestion, the second option was anaerobic digestion. So during anaerobic digestion, microorganisms such as methogenic bacteria break down organic matter into the in the absence of oxygen to produce biogas. So biogas are produced during this digestion process, and the word anaerobic means in the absence of oxygen. So therefore, oxygen is not used in this process, and the bacteria break down the organic matter. So in this, if we put the manure inside and we do not provide it any type of gas, any type of oxygen during this treatment, the microbes convert this manure or organic matter into a mixture of biogases, and these biogases are mixtures of different gases such as methane and carbon dioxide. So these methane and carbon dioxide are then sent for the outlet so that we can use it further in further processing. And the third option in this was composting. And composting is also a microbial process, and during composting, microorganisms such as bacteria and fungus break down the organic matter into humus, which is a nutrient-rich soil amendment. So humus is nothing but a black looking soil which has very rich nutrients in it. So nutrient-rich soil is humus is a nutrient-rich soil which we get uh, with the help of composting. The process of composting also generates heat, which can be used for various applications such as space heating and electricity generation. So, the generation of electricity is generation of heat is a byproduct in composting, and we can utilize that heat for other processes also. So in the right, you can typically see that if the vegetables which we use, if after the waste formation of these vegetables or edible things, we can easily convert them into compost, and this compost can again be used to grow vegetables. So it is also a cyclic process. Now let's move on to the second question. Biogas is produced by degrade organic matter in four general steps by step. So Bioga- biogas is produced in four steps. And the answer the NPTEL showed was hydrolysis, acetogenesis, and methanogenesis. But the correct answer for this is first the hydrolysis takes place, then the acetogenesis, after that acetogenesis, and then methanogenesis. Uh, these terms might be very complicated here, but I will explain this in the next slides. I will explain all these terms. So during hydrolysis, the organic matter, feed the organic matter such as, which contains many different types of compounds such as carbohydrate, protein, and fats. When we feed this organic matter during hydrolysis, the these complex organic matters are converted into soluble matters. So we get what is known as soluble organic matter. So hydrolysis is nothing but a process to convert complex organic matter into soluble organic matter. So first step is hydrolysis. Then comes the second step, which is known as acidogenesis. So in acidogenesis, we get volatile fatty acid. So if we go for the word here, it has acid in it. So the second step is acidogenesis. So in this, we treat these soluble sugars, soluble acids, soluble amino acids, and then convert into volatile fatty acids. And these volatile fatty acids are then converted into some type of acetic acid. So it is known. That's why it's known as acetogenesis. So the name acid here come. You can remember this name as acetic acid. So acetogenesis is a process to convert the volatile organic into acetic acid and CO2. And after this process, we do the methanogenesis. So methanogenesis is nothing but to produce methane. So first we use complex compounds, convert it into soluble compounds, then convert it into acetic acid, and then convert it into methane. So this is a typical chemistry behind the complete process to create methane from complex organic matter. Let's move on to the third question. High OLR organic loading rate would boost the acidogenesis stage of easily degradable substrate and the excess accumulation of what further inhibit the activity of microorganisms. So we know that in acidogenesis we the major thing which we get is volatile fatty acids. We get volatile fatty acids. So accumulation of what substance will inhibit the activity of microorganisms? First is methane. We know that we do not get methane in this process. We will get methane in methagenic process. And we have some other options also: pyruvic acid, lactic acid, and volatile fatty acids. So pyruvic acids and lactic acids are not that highly volatile fatty acids. These are simple acids. So the option is the correct option is volatile fatty acids. So, but how does it uh, inhibit the microorganisms reaction? So, excessive accumulation of volatile fatty acids can inhibit the activity of microorganisms because they can lower the pH of environment. So, if the volatile fatty acids will be higher, the pH of the environment will be lower, and this change in pH of the environment will cause the microorganisms to inhibit their activity. Also, the volatile fatty acids are produced during acidogenesis, which is the phase of anaerobic digestion where complex organic matter is converted into small molecules like fatty acids, simple sugars, and amino acids. When the organic loading rate is high, if this rate is high, there will be more organic matter for the microorganisms to digest, leading to increased production of VFAs. And VFS, if accumulated at very high level, the pH of environment may decrease, leading to acidification and inhibition of microorganisms. So, if we give a very high loading of organic matter, also known as OLR, organic loading rate, if the OLR is very high, then the production of VFAs will be higher. And if the production of VFS will be higher, it will change the pH of the soil. The pH of the soil will change, and which will inhibit the growth and mechanism of microorganisms, because microorganisms also require some type of conditions. So higher amount of VFS leads to lower inhibition. It leads to inhibition of the microorganisms. So now let's move on to the next question. In which phase of anaerobic digestion biomass is converted into smaller molecules such as fatty acid, simple sugar, and amino acid, respectively? So we have discussed this earlier also, that to convert the large organic polymers such as carbohydrate, fats, and proteins to smaller compounds such as soluble molecules, if we need to convert these large into small, then we use hydrolysis, which is the first step. Therefore, the answer is hydrolysis. So, first we will discuss about the what is anaerobic digestion. So, aerobic digestion. So, aerobic digestion is a biological process where organic matter is broken down by microorganisms. Microorganisms are used to break down the matter in the presence of oxygen. So, the first discussion in the previous slide was about anaerobic. So anaerobic means absence of oxygen, but this is aerobic. So this means presence of oxygen. So in an aerobic digestion, microorganisms are used to break down the organic matter into to produce lower organic matters or simple organic matters. It is a common method for treating organic waste such as sewage sludge and food waste. And the phase of organic digestion in which biomass is converted into smaller molecules such as fatty acids, simple sugars, and amino acids is called hydrolysis. We've discussed this in the previous slides also. So, but what is hydrolysis? So, hydrolysis is a chemical reaction in which water is used to break down the larger molecule into a smaller subunits. In simple terms, we just use water to break down large molecule into smaller molecules. And if we talk about the hydrolysis in digestion, the hydrolysis is the initial step where complex organic matters such as carbohydrate, fats, protein are broken down to simple molecules such as simple sugar, fatty acid, and amino acids. This process is usually carried out by the enzymes produced by hydrolactic bacteria. In this phase, enzymes break down the large organic polymer into smaller molecules that can be further processed by other microorganisms in the subsequent phase, such as acidogenesis, acetogenesis, and methanogenesis. So after the hydrolysis, we get the smaller organic polymers, smaller organic matter, and then we can go for further stages such as acidogenesis, acetogenesis, and methanogenesis to create methane gas. So now let's move on to the next question. How many ATP molecules produced during glycogenesis of glucose to pyruvic acid? So there is a little bit confusion here. Glycogenesis is not a process to convert glucose to pyruvic. It is glycolysis. So the question should be how many ATP molecules produced during glycolysis of glucose to pyruvic acids. So the correct answer is here two, and I will first explain what is glycogenesis and what is glycolysis. What is the difference between them? So glycogenesis, which was asked mentioned in the question, is actually the process of converting glucose into glycogen for storage in the liver or muscles. So glycogen is different. It happens inside our body, and on the other hand, glycolysis, which should be the question about it, is a process of converting glucose into pyruvic acid. And during glycolysis, two ATP molecules are produced. So during glycolysis, glucose is broken down into two molecules of pyruvate, and this process is a net two ATP molecules. So this is a typical glycolysis process in which glucose is converted into pyruvic acid and we get two ATP molecules. Now let's move on to the sixth question. Theoretical yield of alcohol from 100 gram of cellulose is, 100 gram of glucose is. So the answer here is 51.14 gram. I'll explain this in the next slide. So we know that the glucose can be broken down into ethanol. So if we go to in the chemical reaction, 1 mole of glucose gives 2 moles of ethanol. So 1 mole of glucose gives 2 mole of ethanol. And if we and what is 1 mole? What is a mole? So mole means gram mass upon molar mass. So 1 mole means now the question had the question has mentioned that how much gram of ethanol will be produced. So by 100 gram of glucose. So if we take glucose in this 100 gram of glucose, so the moles of glucose is 100 by 180. So 100 gram by 180, 180 is a molecular mass of glucose. We can get moles of it. So in case of this, 0.55 moles are used. If we say that 100 gram of glucose is used, it means 0.55 grams mole is used of a mole of glucose is used, and it will produce 2 moles of ethanol. So it means that 1.11 moles of ethanol will be produced. So now we know that what how much amount of ethanol will we get. But 1.11 moles means, so 1.11 equals to gram upon molar mass of ethanol, and molar mass of ethanol is 46.07. So if we multiply this, we will get the answer as 51 point something near about 8. But I have taken this 0.55. If we will take the accurate value of it, 0.5556, and do the calculation, we'll get a value of 51.14. So in this way, we can get the amount of gram require which we will get of it by 100 gram of glucose. So the method to calculate is that we should know the chemical reaction, that how much moles of glucose are making what amount of ethanol. Now let's move on to the seventh question. Clostridium is known for the production of. So Clostridium is a type of species and it's used mainly for the production of butyric acid. So the correct answer is D. So, Clostridium is a genus of anaerobic bacteria that can be found in soil, water, and the digestive tracts of animals and humans. And some species of Clostridium are known for their ability to produce various metabolics such as organic acids, alcohol, and gases through fermentation. These bacteria are also associated with various diseases such as tetanus, botulism, and gas gangrene. So Clostridium is a particular species, and it is used to make organic acids, alcohols, and various gases through fermentation. And majorly, it is used to produce butyric acids. So Clostridium bacteria are known for their ability to produce a variety of organic acid through the process of fermentation. Butyric acid is one of the organic acid that can be produced by this bacteria. During the fermentation process, Clostridium bacteria break down organic compounds such as sugars, carbohydrates, and produce butyric acid as a byproduct. So it breaks down the sugars and carbohydrate to produce butyric acid. The production of butyric acid by this bacteria is an important role, and it plays a very important part in industrial processes including production of solvent and biofuels. So Clostridium is a major species of bacteria which can be utilized to produce butyric acids. Which process is related to humus? So we know that humus is a is a substance which we obtain after it is a type of nutrient-rich soil we studied in the previous slide, and this we get after the composting process. So the correct answer for this is composting. So humus is the stable organic component of soil that gives it its characteristics dark color and helps to improve soil structure, water holding capacity, and nutrient ability. So this is also used and mixed with soil to provide nutrition to the soil. And the process related to humus is composting. Composting is nothing but a simple natural process of decomposition of organic matter by microbe under aerobic condition. The end product of composting is humus, a nutrient-rich soil-like material that can be used as a soil amendment or fertilizer. Composting, and what is composting? Composting is a natural biological process such as that transfer organic waste such as food, scrap, yard trimming, leaves into soil-like substance called compost. So we can use any type of organic matter, such and we can also use waste organic matter such as waste foods, and it is a controlled process of decomposition that is carried out by microorganisms. So microorganisms will degrade these. Microorganisms will degrade these biological organic waste, and during after the decomposition, we can get a nutrient-rich soil, which is known as compost. Composting can be done at home or at a large-scale industrial facility, and the resulting compost can be used as a nutrient-rich soil amendment for plant and gardens. Which gas is common produced along with products during all kind of fermentation process? So during fermentation, there are very variety of gases which we get, and there are very different types of fermentation processes also. But there's one gas which is common and produced every time, and that is carbon dioxide. So carbon dioxide is commonly produced along with products during all kind of fermentation processes. In any kind of fermentation process, we will get carbon dioxide. Fermentation is a metabolic process in which microorganisms such as bacteria or yeast converts organic compounds such as carbohydrate into other compounds. This process is carried out in the absence of oxygen and is used in various industries to produce products such as ethanol, lactic acid, and other chemicals. And some common examples of fermentation process include production of beer, wine, bread making, and some biofuels also. But in almost all the process, CO2 is produced every time. So it is the most common gas which is produced during the fermentation process. And apart from carbon dioxide, various other gases such as methane, hydrogen, nitrogen, and hydrogen sulfide can also be produced during fermentation depending on the specific microorganism and conditions involved. However, CO2 is generally the most common gas produced in most fermented process. Now let's move on to the final question. Which of the following is not the use of baffles? So the correct answer for this is to improve the fermental capacity. So baffles are used in the fermentation process just to improve the mixing. And how do these baffles are work? I will explain this here. Baffles are structure used in tanks or vessel to control fluid flow and increase mixing efficiency. So it will increase the mixing efficiency and control the fluid flow. And they are usually made up of flat plates or panels that are strategically placed inside the tank to alter the flow pattern of liquid or gas being mixed. Baffles helps to prevent vortex formation, improve aeration efficiency, and increase the effect of agitation, which can improve mixing and reduce blending time. They can also help to improve the capacity of the fermenter or bioreactor by creating more uniform distribution of nutrients and promoting better contact between the cells and substrate. So this is a typical baffle which is used. These are mainly flat plates, and these can be used to alter the mixing of the air and the liquid inside the fermenter. Baffles are used in bioreactors to prevent the formation of vortex. So baffles are used in bioreactors to prevent the formation of vortex in the liquid, which can cause inefficient mixing and reduce oxygen transfer efficiency. The use of baffles also improve the mixing and agitation of the liquid in reactor, leading to better mass transfer and nutrient distribution. So better distribution is also happening with the help of baffles. Baffles can also increase the effective volume of the reactor. It also increases the effective volume reactor by reducing the dead space used by stagnant zone in the corners of the tank. So when we mix something in any container, the corners of the tanks, there is not a very good mixing here in the corners. So the use of baffles uh, increase the effective volume of the reactor by reducing the dead space. So these dead space are reduced, and thus they increase the efficiency of the reactor. However, Buffers do not directly improve the fermenter capacity. It is not improving the capacity directly, which is primarily determined by the size and design. So capacity will be determined by the size and design of the fermenter, but it increases the capacity by just utilizing the dead space of the corner of the fermenter. Now we will move on to the assignment 6 and of 2021 and 2022, and most of the questions were of 2023 only, so I have not discussed them here. So let's move on to the first question. Which of the following are the products of fermentation? Lactic acid, hydrogen, ethanol, or all of the above? So during fermentation, we get ethanol as a major, but we get all of these products. So therefore, the answer is all of the above. Fermentation is a metabolic process that converts sugars or carbohydrate into other products through the action of microorganisms such as yeast or bacteria. The products of fermentation depend on the type of microorganisms involved and the specific condition of the fermentation process. In the case of biomass, fermentation can be used to convert complex organic molecules into simpler compounds such as lactic acid, hydrogen, and ethanol. During the fermentation process, microorganisms break down the biomass into simpler molecules by using enzyme to create the reaction. So the breakdown of biomass occurs in very several stages with different microorganisms responsible for each stage. So many types of microorganisms are used, and every type of microorganisms at every stage has a different purpose. So, if we look for a example, in anaerobic digestion, bacteria convert organic matter into simpler molecules such as VFS. We have seen that some type of bacteria are used to convert simple molecules into VFS, which are then converted into methane and carbon dioxide by methogenic bacteria. So we had three different stages such as acetogenesis, and methanogenesis. So all these three stages has different kinds of bacteria, different types of microorganisms, and all of them have different responsibilities to create different products. And in ethanol fermentation, yeast converts sugars into ethanol and carbon dioxide. So majorly in fermentation process, we are using, we are creating ethanol and carbon dioxide from the processes. Now let's move on to the second question. Short HRT may lead to a risk of washout of microbial. Short HRT may lead to a risk of washout of microbial communities. True or false? So the answer for this is true. Yes, these are responsible for microbial communities. But before going in the answer, let me explain you what is HRT. So HRT, it means hydraulic retention time. Hydraulic retention time is the average amount of time that a liquid remains in a bioreactor, uh, or in a wastewater treatment plant, or in any other system. So the amount of time a liquid remains in the reactor is known as HRT. And if the H- and this HRT is an important parameter in biological treatment process, as it determines the amount of time that microorganisms have to degrade or metabolize pollutants in the liquid. A longer HRT allows more time for the microorganisms to consume the pollutants, while a shorter HRT may not provide enough time to complete the treatments and can result in lower treatment efficiency. If this HRT is too short, there may not be enough time for a microbial community to degrade the organic matter into the wastewater. So, the short HRT, in case of short HRT, the lesser time for the microbial community to degrade the organic matter will not result into better efficiency, which will also lead to the washout of microbial communities, which reduces the effectiveness of the treatment process. Therefore, it is an important parameter to optimize the HRT based on the characteristics of the wastewater and the performance of the treatment systems. Which biogas component is responsible for corrosion of the digester equipment? So there are many different types of biogases produced in the digester, and among them, the hydrogen sulfide is the most corrosive gas. So this hydrogen sulfide is responsible damage to the digester equipment. So the correct answer is C, hydrogen sulfide. And hydrogen sulfide is a highly corrosive gas that can damage the equipment used in the biogas digester, including pipeline walls and engines. It is produced as a byproduct during the anaerobic digestion. So in the anaerobic digestion, we get H2S gas, which is all, which is short for hydrogen sulfide, and particularly when the digestion process is not optimized or controlled properly. And if this process is not controlled properly, the production of H2S will be higher. Therefore, the removal of hydrogen gas from the biogas is necessary for safe and efficient use of biogas. So we have to also remove this hydrogen sulfide if the production is high. And how can we remove this hydrogen sulfide? How can we purify the biogas? So this purification is done through a process called desulfurization or biogas purification. So one of the most common method for desulfurization is the use of iron oxide scrubbing. So in the iron oxide scrubbing, the biogas is passed through tank filled with iron. So there will be a tank in which will be filled with iron oxides, and the gas will be passed through it. When the gas will be passed through, the iron oxide will react with the hydrogen sulfide to form iron sulfide, and thus the outcome outlet of the this process will be pure biogas. So other methods of biogas desulfurization includes activated carbon adsorption, biological desulfurization, and chemical scrubbing. But the most common use is iron oxide scrubbing. For ethanol production, which microbe is mostly used? So to use ethanol, the most used microbe is Saccharomyces cerevisiae. So this is the mostly used microbe because these microbes are not resistant to ethanol. So when the ethanol will be produced by these microbes, uh, these three microbes, the rest of the microbes uh, will be get killed with the ethanol itself. So therefore, Saccharomyces cerevisiae is mostly used. The process of ethanol production using S-myrosis involves following steps. So first, we can prepare the fermentation medium, and this fermentation medium is prepared using a carbon source such as molasses, glucose, or and a nitrogen source such as ammonium sulfate or urea. So a combination of carbon source and nitrogen source are used to prepare the fermentation media. So these carbon and nitrogen are the media for the bacteria, and then we need to inoculate the bacteria. These are then inoculated into the fermentation medium and allowed to grow under controlled conditions. So these are further than growth under the controlled condition. And during the fermentation, the yeast cells metabolizes the sugar and produce ethanol and carbon dioxide. The fermentation is carried out under anaerobic condition to prevent the oxidation of ethanol to acetic acid. And finally, we do the distillation for the purification of ethanol. So the ethanol produced during fermentation is separated from the fermentation broth by distillation process. Overall, it is a very preferred microbe for ethanol production due to its availability, efficiency to convert sugars to ethanol, and its high tolerance to ethanolic concentration. Therefore, this microbe is mostly used to make ethanol. Following which are not considered as methogens. So methogens are those microbes which are used to convert the acids into methane gas. So the answer of this is the NPTEL says is autotrophs, but all of these above listed can produce the methane from the acids? So the answer for this is none of the above. Autotroph, methylotroph, acetotroph, and hydrogentroph. All microorganisms are above are listed as methogens. So now let's move on to the assignment 7 of 2021, and we will discuss from here the assignment 7 of 2021 and 2022, and I am not discussing those answers which are repeated in 2023. So only few questions are there, and these questions will help you in determine in solving the assignment 7 of 2023. The fatty acids are frequently represented by a symbol CXY, where X, so CXY. For example, if there we see a question with we see a statement in which it is written that C18 1, so it means that the that fatty acid has a carbon chain of 18 carbons, and there is one double bond in it. So EC181 means 18 carbon chain and one double bond. Therefore, the X means the number of carbon and Y means number of double bonds. So these are what saturated and unsaturated fatty acid look like. Now, if we see the above example, it has, let's count how many carbon does it has. For example, it has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it will represent as C10 0 because it has 10 carbon atom but 0 double bond. It has a double bond of C double bond, but we are looking particularly for C double bond C. It doesn't have any kind of bond here. But in the unsaturated fatty acid, we see that there is one double bond, and number of carbons are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this will be known as C10 1 because it has one double bond. So I hope you can now tell by looking what CXY means. Now let's move on to the next question. Acid esterification is well accepted as a pre-step of the base catalyzed trans-esterification reaction to esterify FFA if. So acid esterification is a process to convert acids into esters. So the question is about pre-step for the base catalyzation reaction to esterify if higher, lower, equal or none of the. So the correct answer for this is higher than 2%. So esterification is usually considered as a pre-step for the base catalyzed transition to reaction to esterify free fatty acids. So before going to the trans-esterification, we do the esterification, and it should be more than 2%. If the free fatty acids content is lower than 2%, it is generally not necessary to perform acid esterification as a base catalyzed transformation reaction can acidify both triglyceride and FFA. So, what is esterification? Esterification means to convert free fatty acid into ester, fatty ester, and after this esterification, we do the trans esterification, and in this we again get a ester but a different. This carbon is replaced by some other carbon. So before the trans esterification we need to do the esterification, but the amount should be higher than 2% of free fatty acids because these free fatty acids are only converted during this process, so these must be greater than 2% of the amount. The main disadvantage associated with the biodiesel purification by wet washing method are, so when we do the purification of biodiesel, we can do it by various methods. In one process, we use wet washing. This wet washing has some kinds of disadvantages. So, first is it emulsion formation, and second is drying of the final product. So, the correct answer is both A and B. Both are disadvantages to it, and I will explain this in the next slide. When uh, first when we take the biodiesel is oily in nature, and during the wet washing we use water. So when we mix these oil and water, we get something which is known as emulsion are some kinds of compound in which water and oil are mixed, such as creams which we use. So the so during the wet washing of the biodiesel with the water, emulsion formation can occur. So these emulsion formation occurs during the mixing of water and biodiesel, which leads to the formation of stable emulsion. These emulsions are also very stable. The problem with emulsion is that if we do the washing and the biodiesel and water get mixed very uh, homogeneously, then we cannot get biodiesel from it. The separation of water is very difficult. So if we need to separate the water or it is a very energy intensive process, and process such as evaporation or vacuum distillation will be used to remove the excess water from the biodiesel, which will also improve the cost. Therefore, both are the disadvantage. First is emulsion formation, and second is leading to high energy usage. So wet washing is usually not used to purify biodiesels. In fluidized bed reactor of FTS, attrition resistance catalysts are used because. So the correct answer is that high temperature is associated in this process. I will explain this FTS in the next slide. That what is this FTS? So FTS stands for Fischer-Tropsch synthesis, which is a process of converting carbon-based feedstock such as coal, natural gas, biomass into liquid hydrocarbon. So it is a process in which we take biomass and then convert it into fuels, and these fuels are majorly liquid hydrocarbons. These are not gases. Majorly, they are liquid hydrocarbons. So when this in this process involves a series of chemical reactions that produces hydrocarbon, and we get hydrocarbons of various chain length and structure by combining carbon monoxide and hydrogen. This process was first developed by French Fisher and Hans Stokes in 1922 and has been since refined and improved for various chemical processes. And this process of converting the biomass into liquid fuel involves very high temperature and pressure. So, FTS is a high temperature and high pressure process that requires catalysts to be maintain their activity and selectivity over a long period of time. So it also requires catalyst, and these catalysts should have their activity higher at long period of time. The use of attention resistant catalyst ensures that the catalyst do not break down or become deactivated, which can lead to decrease in the yield of desired product. So these catalysts are typically made from metals such as iron, cobalt, nickel. So these catalysts are mostly metals, so that the efficiency is increased over time, and these are also resistant to most chemicals. And these metals is also used as a catalyst because FTS is a very process in which involves very high temperature and high pressure. So now let's move on to the next question. The lowest temperature at which the fuel still maintains its fluidity is known as. So there are four different types of temperatures: cloud point, pour point, cold filter plugging. But among this, pour point is the lowest temperature at which the fuel will have its fluidity. So, but what are these four different temperatures? What are these four different? What is cloud, pour, and cold filter? So, first we'll discuss about the pour point. Pour point is the lowest temperature at which fuel or oil will still flow or pour, as the name has pour point. So in this, it is a temperature at which fuel can have fluidity. But below this temperature, the fuel becomes more viscous and may even solidify, making it difficult to handle and use. So majorly all the fuels are used above the pour point. Cl- Now let's move on to the cloud point. Cloud point is a temperature at which wax crystals begin to form in the fuel. So if we go below the pour point, the fuel will become very viscous, and after this viscous formation, it will solidify. During this solidification, there will be crystal formation in the uh, fuels, and this crystal formation is happens in the cloud point. So these wax crystal can cause breakage in the filters and fuel cell, leading to engine problems. Therefore, the fuels are not used at cloud point. And then it's the next point was cold filter plugging point. Cold filter plugging point is a temperature at which fuel begins to clog the filter. At this temperature, the wax in the fuel begins to solidify and can block the fuel filter, reducing fuel flow to the engine. So every time, if we want to use any type of uh, biofuel, we need to use it above the pour point. So that the fuel may have fluidity in it. What the disadvantages associated with acid transfer transesterification is? So what is acid transesterification? In this, if we have let's say we have some kind of ester which is represented by RCOOR, and we use some kind of acids such as RCOOH and do the reaction in it. In this, we get a new ester which is represented by RCOR dash. So this is known as transesterification process. There are many disadvantages of this, such as slow reaction speed, high temperature requirement, and difficult glycerol recovery. So therefore, the answer is all of the above. Now let's move on to the assignment 7 of 2022, and this assignment, the questions discussed here will help you in answering the assignment 7 of 2023. So first question for this, and I have not discussed all the questions of this year, as it most of them were repeated this year also. So the second question of assignment 7 was higher iodine value and saponification value indicate. So higher, whenever you see that if a thing has higher iodine value, it will have higher degree of unsaturation and lower molecular weight corresponding to vegetable oils. I will explain this in the next slide, that what does this mean. So iodine value and saponification value, these two are some kind of quantitative analysis of vegetable oil, and this can provide us about the chemical properties of the vegetable oils. The iodine value measures the degree of unsaturation. So, and this degree of unsaturation indicates number of double bond. So higher iodine value indicate higher degree of unsaturation. So if we see somewhere that it has a very high iodine value, it means that the unsaturation bond will be higher. Therefore, the fatty acids present in it will have high amount of double bonds. So you can see that if a if a type of oil has C18(2) or C18(1) written, so it means that it has two unsaturated bond and one unsaturated. There is one double bond and two double bond in. And if these are very higher, in C18(0), it is lower, then it may have high iodine value, because iodine value represents unsaturated bonds. And second is saponification value. Saponification value measures the molecular weight of fatty acids in the oil. Higher saponification value indicates lower molecular weight of fatty acids in the oil. So saponification is, if it is higher, the molecular weight will be lower. Molecular weight of fatty acid in the oil will be lower. This is because saponification process is a process in which the breakdown of fatty acid into smaller components such as glycerol and soap, and the amount of alkali required for the reaction is used. So if the higher amount of oils if higher amount of this materials are used to break down, then the molecular mass of the fatty acids present in it will be lesser. Now let's move on to the third question. Palm oil is more saturated than soybean oil and rapeseed oil as its major fatty acids include. So by now we have seen that what does this C16 C18 means. That C16 means that it has 16 carbon chain, and C16 means it has 16 carbon chain and one double bond. So in a same zero zero double bond if it is zero, and if it is C16 it means it has 16 carbon chain and one double bond. So palm oil is more saturated than soybean and rapeseed oil. So saturation means that it has less amount of double bonds. So palm oil has less amount of double bonds than when compared to soybean and rapeseed oil. So the question was this: what is the major fatty acid which palm oil has? So palm oil, the major component is, first is palmitic acid, and the second major component is oleic acid. So palmitic acid and oleic acid are the major components of palm oil, and stearic acid and linoleic are also the components of it. And it has more zeros than ones and twos. So the chain involves higher amount of C16 zeros and C18 zeros when compared to C18 one or C18 two collectively. Therefore, this is more saturated. The palm oil is more saturated than soybean oil and rapeseed oil. On the other hand, soybean oil has a very high amount of double bonds. It has C18 one, C18 two, and C16 one also. It has a very high amount of unsaturated oils, and rapeseed oil also has very high amount of double bonds in it. So palm oil has mixture of unsaturated, oh sorry, saturated and unsaturated. So it is more saturated than rapeseed and soybean oils. Now let's move on to the next question. Magnosol is one of the commonly produced available silica absorbent used for biodiesel purification. So we have seen that biodiesel purification we can do with. In the previous question, we have discussed that purification can be done by washing, also known as wet washing. But it had many disadvantages such as emulsion formation and high heat requirement to remove the water from it. And there is another method to purify the biodiesel is to use some kind of adsorbent. This adsorbent, when we pass the biodiesel or biofuel through these adsorbent will stick the impure material onto it. And these adsorbent are mostly based on silica. So magnesol is the most used commercially available adsorbent, which we can use for the purification of biodiesel. But what is this magnosol? So the question is, it is an inorganic mixture of which of the following compounds? So first of all, we can easily guess from the name magnosol. There is a word Mag in it, so it represents magnesium. So it is a mixture of magnesium silicate and sol. Sol here means sulfates. So it is a mixture of magnesium silicate and anhydrous sulfates. So magnosol is a commercially available adsorbent used for biodiesel production. We have seen this, and it is made up of magnesium silicate and anhydrous sodium sulfate. Hence the name magnosol. And it is and magnosol is used to remove the impurities present in the biodiesel, such as soap, glycerides, and other polar compounds from the biodiesel. And how does this happen? How is this purification takes place? The process of biodiesel purification using magnosol involves mixing the adsorbent with a crude biodiesel. So we will mix the adsorbent with a crude biodiesel. Crude biodiesel means impure biodiesel, and then filtering the mixture to remove the adsorbent. After and during this mixing of this crude biodiesel and absorbent, the impurities will be physically adsorbed on this magnosol, and then we can use a filter process to remove this adsorbent. So the adsorbent adheres the impurities which becomes trapped within the magnosol. The resulting purified biodiesel is then free of impurities and can be used as a fuel source. So basically it's a adsorption phenomena, and it's made up of magnesium silicate and sodium sulfate. Now let's move on to the next question. For acid esterification of feedstock with higher FFA, what is the most effective catalyst? So FFA is a short form of free fatty acids, and we have seen in the past slides also the what are these free fatty acids. These are C16 1, C16 0, C18 1, C18 2. All these short forms are free fatty acids. And what are these C16 C18? These are nothing but C18 means that there is a 18 carbon chain with at least with two double bonds. So it means that it is C18 2. And at last it has COOH. And this COOH represents acids. So these are free fatty acids. So to esterify these acids, we need to esterify this COH to make fatty ester of it. To convert the fatty acids to fatty ester, we need some types of catalyst. So the question is that what among this is the most effective catalyst? So the answer is hydrochloric and sulfuric both. Actually, both of these acids can be used as a catalyst for free fatty acid, but sulfuric acid is mostly used because HCl is more corrosive in nature than sulfuric acid. And this formic acid and acetic acid are not used. These can also act as a catalyst, but these two also produce some kinds of bioproducts which are not desirable. So sulfuric acid is mostly used during this esterification reaction. So sulfuric acid is more effective catalyst for acid esterification of feedstock with higher FFA because it has a higher acidity and is stronger proton donor than other acids. This allow it to catalyze the reaction more efficiently and with higher yield. Hydrochloric, formic acid, and acetic acid, the rest three options which were mentioned, can also be used as a catalyst, but they generally are less effective than sulfuric acid. These are less effective. Also, hydrochloric acid is corrosive in nature. So there are many safety concerns while using HCl. And while formic acid and acetic acid will lead to the formation of impurities in the biodiesel products. Therefore, acid is only preferred because HCl, formic acid, and acetic acid has other disadvantages. The four metals considered for FTS is so FTS we have seen that is a process which involves high temperature and high pressure, and thus the catalyst used for the FTS are mostly metals, so that it have uh, higher longevity, and it can also be it can also perform better in high temperature and pressure. So, but what are these metals? What are is the composition of these metals? So, majorly there are four metals which are considered for FTS, and that is iron, cobalt, rhodium, and nickel. So, FTS stands for Fischer-Tropsch synthesis, which is a chemical process to convert carbon-based sources such as coal, natural gas, biomass into biofuels. This process involves breaking down the carbon feedstock into smaller molecules called syngas and then using a catalyst to combine these molecules. The choice of catalysts can vary, but iron, cobalt, rhodium, and nickel are the most commonly used. So these four are the most commonly used catalysts for the Fischer-Tropsch synthesis. FTS is an important process for producing synthetic gas, especially in areas where traditional oil reserves are limited. So these four iron, cobalt, rhodium, and nickel are the mostly used metals used as a catalyst in the FTS process. So uh, we have discussed today about the assignment 6 and assignment 7, and I hope these assignments of 2021, 2022, and 2023 of assignment 6 and 7 will help you in doing the assignment 7 of this coming week. And I wish you all the best for the assignment, and we will meet you next week where we will discuss the assignment 8 for the session.